Hello friends, here is our new video on carbohydrates. Let's start. Most carbohydrates are polymers. Most carbohydrates, as well as proteins and nucleic acids, are polymers. What is a polymer? Polymers are large, complex molecules composed of long chains of monomers joined together. Monomers are small, basic molecular units. Examples of monomers include monosaccharides, amino acids and nucleotides. Carbohydrates are made from monosaccharides. All carbohydrates contain the elements carbon, hydrogen, and oxygen. The monomers that they're made from are monosaccharides, for example glucose, fructose and galactose. Glucose is a hexose sugar, a monosaccharide with 6 carbon atoms in each molecule. There are two types of glucose, alpha and beta. They're isomers. Isomers are molecules with the same molecular formula as each other, but with the atoms connected in a different way. You need to know the structures of both types of glucose for your exam. It's pretty easy because there's only one difference between the two. Condensation reactions join monosaccharides together. A condensation reaction is when two molecules join together with the formation of a new chemical bond, and a water molecule is released when the bond is formed. Monosaccharides are joined together by condensation reactions. A glycosidic bond forms between the two monosaccharides as a molecule of water is released. A disaccharide is formed when two monosaccharides join together. Example: two alpha-glucose molecules are joined together by a glycosidic bond to form maltose. Sucrose is a disaccharide formed from a condensation reaction between a glucose molecule and a fructose molecule. Lactose is another disaccharide formed from a glucose molecule and a galactose molecule. One tip for exam: if you're asked to show a condensation reaction, don't forget to put the water molecule in as a product. Hydrolysis reactions break polymers apart. Polymers can be broken down into monomers by hydrolysis reactions. A hydrolysis reaction breaks the chemical bond between monomers using a water molecule. It's basically the opposite of a condensation reaction. For example, carbohydrates can be broken down into their constituent monosaccharides by hydrolysis reactions. Use Benedict's test for sugars. Sugar is a general term for monosaccharides and disaccharides. All sugars can be classified as reducing or non-reducing. Benedict's test tests for sugars. It differs depending on the type of sugar you are testing for. Reducing sugars include all monosaccharides, for example glucose, and some disaccharides, for example maltose and lactose. You add Benedict's reagent, which is blue, to a sample and heat it in a water bath that's been brought to the boil. If the test's positive, it will form a colored precipitate, solid particles suspended in the solution. The color of the precipitate changes from blue to green, to yellow, to orange, to brick red. The higher the concentration of reducing sugar, the further the color change goes. You can use this to compare the amount of reducing sugar in different solutions. A more accurate way of doing this is to filter the solution and weigh the precipitate. If the result of the reducing sugars test is negative, there could still be a non-reducing sugar present. To test for non-reducing sugars, like sucrose, first, you have to break them down into monosaccharides. You do this by getting a new sample of the test solution, adding dilute hydrochloric acid and carefully heating it in a water bath that's been brought to the boil. You then neutralize it with sodium hydrogen carbonate. Then just carry out Benedict's test as you would for reducing sugar. If the test's positive, it will form a colored precipitate, as for the reducing sugars test. If the test's negative, the solution will stay blue, which means it doesn't contain any sugar, either reducing or non-reducing. Tip for the practical test: always use an excess Benedict's solution. This makes sure that all the sugar reacts. Polysaccharides are loads of sugars joined together. A polysaccharide is formed when more than two monosaccharides are joined together by condensation reactions. For example, a lot of alpha-glucose molecules are joined together by glycosidic bonds to form amylose, you need to know about the relationship between the structure and function of three polysaccharides: starch, glycogen and cellulose. Starch is the main energy storage material in plants. Cells get energy from glucose. Plants store excess glucose as starch. When a plant needs more glucose for energy, it breaks down starch to release the glucose. Starch is a mixture of two polysaccharides of alpha-glucose, amylose and amylopectin. Amylose, a long, unbranched chain of alpha-glucose. The angles of the glycosidic bonds give it a coiled structure, almost like a cylinder. This makes it compact, so it's really good for storage because you can fit more into a small space. Amylopectin, a long, branched chain of alpha-glucose. Its side branches allow the enzymes that break down the molecule to get at the glycosidic bonds easily. This means that glucose can be released quickly. Starch is insoluble in water and doesn't affect water potential, so it doesn't cause water to enter cells by osmosis, which would make them swell. This makes it good for storage. Use the iodine test for starch. If you do any experiment on the digestion of starch and want to find out if any is left, you'll need the iodine test. Just add iodine dissolved in potassium iodide solution to the test sample. If there is starch present, the sample changes from browny orange to a dark, blue-black color. Make sure you always talk about iodine in potassium iodide solution, not just iodine. Glycogen is the main energy storage material in animals. Animal cells get energy from glucose too. But animals store excess glucose as glycogen, another polysaccharide of alpha-glucose. Its structure is very similar to amylopectin, except that it has loads more side branches coming off it. Loads of branches means that stored glucose can be released quickly, which is important for energy release in animals. It's also a very compact molecule, so it's good for storage. Cellulose is the major component of cell walls in plants. Cellulose is made up of long, unbranched chains of beta-glucose. When beta-glucose molecules bond, they form straight cellulose chains. The cellulose chains are linked together by hydrogen bonds to form strong fibers called microfibrils. The strong fibers mean cellulose provides structural support for cells, for example in plant cell walls. Here are some practice questions. You can give answers in the comment box and we will review it. Thank you for watching video. Please share with your friends and family and subscribe to the channel Biology Notes. If you have any questions regarding biology, please comment below. You can also join us on social media, links are in the description.